Oh, sort of microbes. Yeah, well, I will not be here. Check it out.
Sheree. How are you? It looks like Sheree. I've been trying to do a lot of
No, I didn't get it. All right, everybody can take a seat and we can get started. It's a great pleasure to introduce Sheree Watson. Sheree has messed it up. Sheree Watson, who is part of the Ike Wai project here on groundwater resources in Hawaii. She started in October 2017, so she's here slightly more than a year, 1.5 years now. And she's been working on microbes primarily. And just like Kiana, she's super excited about what these little creatures can tell us on connectivity of water, distributional flowpaths, and so forth. Before she joined UH, she did her dissertation work at the Oregon Health and Science University in the Institute of Environmental Health over there, and she focused on the biogeochemical cycling of phosphorus in the Columbia River estuary. So she's always been looking at these kinds of water quality issues so far, and today she will enlighten us with what the microbes can tell us. So Sheree,
great. Thank you.
I get it, sorry.
No, you're fine.
I'm just slow.
Great. Okay. That's the first one, I think. Thank you for coming today, and taking some time, giving me the opportunity to talk about my work today. I am a postdoctoral researcher. I've been here a little over a year, and had the great opportunity to work in the Frank lab with Kiana and her whole lab crew, Frank. Today I'm going to be talking about the work that we've been doing in Kona on the Big Island, and in particular, and I'll talk about this, some of the questions that are being driven by the Ike Wai project and how we're utilizing microbes to try and answer those. So this little diagram here represents going from microbial communities to trying to ask some questions about what they're telling us about the aquifers, and the last one, of course, is a map of the aquifer. So first couple of slides are some maps to just orient ourselves with where we're at. We're in the Hualālai watershed in particular. This is a semi-arid environment. There's no superficial water in this area, and so all of the freshwater resources come from groundwater. It is the aquifer system, which I'm starting to learn more about because I'm not a geologist, is very complex, hard to study, difficult to understand, how voluminous it is, how much water it can hold, how old the water is, how connected it is, are all big questions that are still yet to be answered, and that, you know, as a group of people we're working on. So, in particular, this is a map of the same area, Hualālai, but mapped here are the traditional boundary lines for management of water, ahupua'a. And these lines were put on and really mapped out areas of management according to farming, what they were going to farm in these areas, and just the water availability, right? And if you look, previously, Hualālai volcano there is in the center, and then Mauna Kea and Mauna Loa there for reference. But in the lines that are provided here, you can really see that they run from Hualālai, the volcano, all the way down to the coast. And those were done for political reasons. Water's a very protected and sustained resource there. And so these lines were drawn for water availability, also what they could farm in those areas, and access to the coast, and some of the alkaline pools that occur out of the coast, and some of the freshwater sources that occurred along those areas. So traditional look at Hualālai. Now we flash forward to more of a modern look at this watershed area. Same area, I've labeled the two aquifers that occur in Hualālai, Kiholo and Keho. And these maps, I superimposed two maps on top of each other. The colors refer to private land ownership in this watershed area. And then the map on top of that is the map of cesspools and septic systems in those two aquifers. And what you can see here is there's quite a few dots in the Keho aquifer here, and those are all septic systems or cesspools, and then very little of that in the Kiholo aquifer. So quite a difference, and those are indicative of like urban development, right, and people. And so that's very representative of probably inputs into groundwater there. So a lot of difference between these two aquifers. And I think it's also important to point out how private land ownership, how different it is than the ahupua'a boundaries I showed you in the previous map. These areas are not bound by any of the water and planting restrictions that were done previously by traditional Hawaiians. I think it's also important to note, I've learned a lot during this project about Native Hawaiians' traditional use of this area from the Hawaiian newspapers that have been translated, and they talk a lot about, the stories in those papers talk a lot about freshwater resources, caves and freshwater springs that are present there, and how important those are to just living in this area. Okay, so modern, both a traditional and a modern look at this watershed. That leads me to talk a little bit about the Ike Wai project, and Ike Wai means knowledge of water. And that's really what we're seeking to understand here, in particular in the Hualālai watershed. The questions that are being asked are driven by the community stakeholders in the area, and that includes farmers and ranchers, business owners, environmental land stewards, land managers, national parks, some of the Board of Water Supply people. All of those people have gotten together and have put together some things that they don't know about this watershed. So here's some of the examples of the questions. What's the degree of connectivity between high level and basal aquifers? What's the volume of the Keho aquifer? How does the Hualālai volcanic riftstone affect groundwater flow and quality within the aquifer? What's the most cost-effective way to meet future water consumption needs from the Keho aquifer? So as you look at these questions, and then I try to overlay what I know about microbiology, there are some questions here that I know microbes can't answer. I think microbes are incredible, but I know for a fact that we probably can't find a microbe that can answer the question of what the volume of the Keho aquifer is, right? It's just not something microbes can do. I think they can do a lot of things, but there's some of these questions we can't work on alone, but that leads us to the group, this Ike Wai project, and it really is an interdisciplinary, integrated project working across lots of different disciplines to answer these questions. So here Kiana and I are up in the microbiology section, but what we put together, the questions that we try and answer will go into, be integrated into data that will try and answer those questions with all these different groups. So geology, geophysics, Hawaiian translations, which I mentioned before, social science, hydrological modeling, economics. So all of these pieces together, our data all together will try to put together a picture of what's going on in this Hualālai watershed. But I also want to state too that the work that we're doing in microbiology is really based on some preliminary work that's already been done in this watershed, and particularly in geology and in geochemistry. And so I'll talk about that. Those fields have set up a structure for which we're asking our questions, and then hopefully the data that we return on this will go back in and further more research into these areas. Okay, so our hypothesis is really based on previous work that's been done in groundwater systems. and particularly with microbial community diversity. So this is a, let me orient you with these plots first. These are called NMDS plots. The axes are really arbitrary, but what they're trying to do here is visualize microbial communities in space. So when you see this is a sample, a microbial community sample, and when you see two that are close together, that's because they're more similar. With whichever they've chosen here to do to assess relationships, in this case, these two were placed closer together because they're more closely related. Okay, so in this study, they sampled aquifers that were younger in age, newer. They were closer to the surface, had more recharge and more active oxygen cycling in the environment. Compared to the red samples, sorry, lost pointer. The red samples come from older aquifers, deeper at depth, less recharge, less oxygen. So when they plotted these communities on an NMDS plot, what you see is somewhat of a separation between the new microbe communities come from newer aquifers or closer to the surface and older watered aquifers. And then plotted on top of this are concentrations of nutrients and major ions in the groundwater. And they really point out the microbes that are basically interacting with these concentrations. So for example, on one side of this plot, you see iron and sulfate levels. And the microbes that are lining up over here are iron reducers and sulfate reducers. And they're more prevalent in the aquifers that are older in age, less oxygenated. Okay? On this side of the graph, on the opposite side, you see high levels of nitrate and oxygen. And these microbes are denitrifiers and aerobic metabolism organisms. And they're lining up on this side in the newer aquifers. So what this plot is telling you is that microbes react to their environments, right? In this case, it's the amount of oxygen getting into this environment, which is related to where they occur at surface or at depth. And then the microbe's metabolism is basically telling you what's going on in that environment. So this is a kind of a typical way to utilize microbes to tell you what's going on in these environments you can't see, that are difficult to sample. And we use this as a hypothesis for our work in Hualālai. And it's commonly done. Microbes and geochemistry are often done hand in hand. But what's novel and unique about what we're trying to do is we're going to use geochemistry as well, but we're also trying to ask about some geological features and whether or not those will isolate and create similar communities that are different from each other based on their connectivity or lack of connectivity. So this really leads to the questions that we think we can answer as far as the Ike Wai project goes. And this first one is, what is the degree of connectivity between high level and basal aquifers? So in the map over here of the Hualālai watershed again, Hikolo here to the north, Keaho to the south, there is a divide that occurs right here based on the wells that have been put in here. And so these blue dots refer to the groundwater wells that we've sampled. And then I just colored the low level aquifer or the basal aquifer blue and the high one red. But we're looking at communities across this divide to see if we can see differences or similarities within them and infer connectivity between these two. Okay. The second question is a question about the rift zone that occurs between Hiholo and Keaho. It's a volcanic rift zone. It's colored here with red, with a red dashed line. And we're asking if, how does the Hualālai volcanic rift zone affect groundwater flow and quality within the aquifer? So within this Hualālai aquifer, how does the connectivity between these two, how does it occur? Does it occur? Is that actual rift zone impenetrable? Doesn't allow exchange of water? And then try and assay water quality based on that as well. Okay, and then the next question is really based on some of the geochemistry that's already been done in the watershed. And that has to do with oxygen isotope geochemistry that's been performed on submarine groundwater discharge out here at the coast. And the samples here are colored, where samples have been taken are colored here in dots. And based on the oxygen isotope signature out here, the proposed path of where that water originated from rainwater or groundwater then is conveyed in the black lines here, okay? And so you can see this work proposed 5 distinct flow paths within this aquifer or watershed. And the first one here is the orange one up north, red, green, or sorry, blue, green, and purple. And so what we have done is we're using these proposed flow paths based on geochemistry to ask whether or not the communities that we're sampling for are distinctly different in these flow paths. So in particular, we're focused on the Keaho aquifer, which is right here, and you can see underneath the flow paths for blue, green, and purple. We're going to look to see if community diversity analysis can see whether or not these flow paths are distinct from each other. So in the Keaho aquifer, what is the nature of the geological structures is linked to subsurface water storage and flow? So those are the 3 questions we're proposing to answer based on our microbial diversity. I want to talk a little bit about how we're sampling and the process, so you understand how we get to some of the results I'm going to show today. We've been sampling quarterly in the Hualālai watershed, and here again is a map of, in particular, the 2 aquifers in Hualālai, and then the surrounding border aquifers around Kiholo and Keaho. And we have a few sample in these, a few groundwater samples, not as many as we have in these, but these are important because they serve as outliers, right? Comparisons to what you're finding in here, and they can serve as a comparison for what you're finding. So we've been sampling quarterly for the last a little over a year, every 3 months. We have 129 unique sites that we've sampled from. That includes groundwater wells and alkaline pools. 31 different organizations that we work with, and that's really that's both unique and that because you saw the private land ownership map before. It's unique that many different organizations are working with us, and that really goes to Kiana's relationships in this area as well as Greg Chun's and their collaboration with us to work on water problems that everyone is dealing with. So, and those are organizations that take time to continue to keep your relationships healthy with. So when we go out and sample, Diamond and I typically start a month to 2 months in advance of scheduling, because you can imagine what it's like to schedule over 31 organizations within 2 weeks to sample these groundwater wells. It takes us about 2 weeks, and I think the last time we went out, we drove over 1,000 miles to get all these samples, and that includes sampling for Diamond's rain buckets as well. When we go out in the field, we go out as a team so that what we collect goes to many different pieces of the project. We've collected over 1,000 samples, and then really, for microbiology, that's kind of the easiest part of the whole thing. We got to come back, and we tend to spend the next 3 to 6 months processing those samples in the lab, sending them away for sequencing, and then the analysis, the bioinformatic analysis on the other side. So again, the map of the aquifer, these are the sample sites I'll be referring to, and you'll see this map again in different sections. And then again, you can see the high-low divide and the rift zone that we're asking questions about. So this is just a quick pipeline view of what we do. Each of these different well samples each of these different wells are samples that we collect. We usually spend all morning when we're out there collecting about 10 liters of water, and then we bring it back to the NELHA labs where they graciously let us some lab space. We filter, and then each of those samples gets filtered onto a 0.8 filter and a 0.2 filter because we're looking at both free and particle-associated microbes. And then we take them back to our lab at Kewalo. We extract DNA, prepare PCR, barcode all of these samples, and then they get sent away for sequencing. They come back as huge data files, and then we move them through this pipeline so that we can get taxonomic identification of everyone who is in on our filter who started out in the well. Yes? Yeah, one slide for about 3 months of work. That's the way it's goes. Okay, so phylogenetic diversity analysis. I'm going to talk really primarily today about beta diversity. And when I say beta diversity, that's the comparison of microbial communities at between two different samples. So in this case, maybe a well site A and a well site C. If you can also look at diversity as a function of just within site, how species rich is that site, and that's alpha diversity. We're looking at that too, but today I'm going to focus on beta diversity, and that's probably kind of obvious. We're looking for differences between our groundwater wells. So I'll focus on that type of diversity today. I'm going to talk about two different algorithms, matrices in which we analyzed for diversity within these samples. So again, if you go back to the samples that come from the wells we collected for, a Bray-Curtis matrix refers to looking at the differences between communities just based on who is there and who is not there. It's a presence-absence matrix. So in this case, from your original sample, you would get your taxonomic sequences, and you would, using this algorithm, you would just look at who's there and who's not there between samples. That would then be conveyed into a visualization, like I showed before, an NMDS, and samples that are more closely related would show up closer together versus those that aren't would occur further apart on this plot. So Bray-Curtis is simply presence and absence. We're going to compare that to a unifrac, a distance matrix, which is basically a calculation of relatedness based on phylogenetic relatedness. So how closely related are your microbes to each other, and then conveying that into relatedness by diversity. So in this case, it actually calculates the distance in a tree. You create a tree, and it calculates the distance in this tree between relatedness, and then you convey that into an NMDS plot. Okay, so two different ways to look at diversity within our communities, one based on how they're related to each other, and just one based on who's there and who's not there. So here's some of our first data plots, two NMDS plots, and you can see up at the top, I've tried to label, there you go. I've tried to label, I'll always put the phylogenetic analysis on the left and the presence absence on the right, just so that this will be the same all the way through. The NMDS plots here, you can see all the samples are colored. This first look at beta diversity was using season, when we collected these samples. So November is the green and August is blue. And one of the things we were concerned about, and we're not sure in our analysis, was whether or not we would see any seasonality during our quarterly sampling. And that went into the design. We were sampling quarterly to just see if we might see some differences there. So when you look at an NMDS plot, you're looking for patterns. You're looking for samples to group together or not group together. So when you look at this plot, it's pretty obvious that your November samples are grouping together compared to your August samples. Yes? And that indicates that the community structure is probably being driven to some degree by the season that we collected them. And so this is a pattern that we want to pay attention to as we work through the rest of the data. So I will continue to shape the data in the next few slides by when they were collected. You'll see November will always be these star shapes, and August will be the square shapes, just to show that some of the patterns that we're observing here may not always be driven by some of the questions we're asking, but maybe some of the seasonality that we're seeing in our samples. This is kind of unusual and maybe not necessarily expected. This is groundwater aquifers deep underground, right? And you wouldn't expect to see a seasonal signal in some of this, but it's important to note that as we move forward, we have 2/3 of our samples are in the pipeline for analysis. And so we will have a lot more data in the next few months that comes through, and this we'll be able to see if this seasonality pattern continues on through or is just an anomaly based on the 2 sample times we have here. But important to note as far as patterns go. Okay, this next slide is displaying salinity as a variable that we also collected for. So I kept the shape in there so that you can do, but these are colored by the salinity of the wells, the salinity present in the samples that we had. The saltier samples are red, the brackish samples are purple, and the freshwater samples are blue. And so what you see here, if you're looking for patterns, is you see the salty samples grouping together, and then particularly in presence absence in Bray Curtis, and then you see a natural kind of gradation to purple and then blue. So community structure here is being driven by the salinity in the water that we sampled and that they occur in. To some degree, you see the same trend over this. Over here, there's some overlap in some of the brackish and the freshwater samples. So salinity is also a driving factor for community structure within these samples, good to keep in mind. And then the last one we checked for here is, I told everyone we were filtering on a 0.2 and a 0.8 filter, so free or particle associated. And you can see here, these are colored by free. This is a 0.2 filter, so those are red, and then blue is particle associated. And in both of these plots, you do not really see a differentiation between whether or not something was free in the water or particle associated. So as far as we can tell, there doesn't seem to be a differential or a different community structure based on particle or free living in groundwater systems that we're looking at thus far. Okay, so I just want to explain some of the statistical analysis that we used alongside these NMDS plots. You see analysis of similarities, ANOSIM statistics used a lot with NMDS, so that visualization can also be conveyed into statistical significance. An analysis of similarity is a non-parametric statistical test. It's somewhat like analysis of variance, except for it works on distance matrices. So it's an analysis of variance working on the results of the Bray Curtis or the unifrac distance matrix. So phylogenetic relatedness or presence or absence. So often what you will see when you see an NMDS plot with statistical significance is these colored circles where they've tried to, based on this NMDS, these two communities would be grouped separately and would be statistically different from each other. And so often with ANOSIM, you'll see a p-value, a statistical significance value, and an R value. And I'll tell you a little bit what that relates to as well. I just wanted to introduce this concept as I move through our questions so that you can see some of our statistical patterns. Okay, so this is our very first question. Do we see connectivity between the high and low basal aquifers here? And so again, I've colored the high samples red to represent the high aquifer here, and the low samples are blue down here. And the actual groundwater wells, again, are the blue colors here. So in the two plots here, let's start with Bray Curtis, the presence absence. The ANOSIM p-value is significant, but the R value is low. And what R represents is it's a measure of the similarity between the communities. So in this case, the difference between the communities is statistically significant, but when you actually look at the R value, which tells you similarity, these communities are still very similar to one another. And that's different than the unifrac, the phylogenetic comparison. Here, there was no statistical difference between communities, and the R value was also very low. So what does this tell us? If we were to interpret this, just this on visualization, you don't really see a separation of red and blue here, right? They're overlapped, and they tend to group along the same lines as we saw before, seasonality and salinity. So you could infer that these two communities are connected and that they're not being separated by this high-low divide. However, the fact that in the separation, a difference between high and low, may infer that geochemically, these communities are different in the high and the low aquifer. Yes, there's some, it looks probably pretty obvious that there'd probably be some strong seasonality or salinity differences between the high and low aquifer, and those salinity differences may be driving the difference between the high and low in this plot. Okay, so that's our first question. The second one, how are communities similar across the Kiholo and the Keauhou aquifer? So here in this NMDS plot, I've colored by the samples where the samples were collected from. You want to focus in on the green color and the purple color here, because that's the Keauhou is green and the Kiholo is purple. And if you look over in the NMDS plot, you can see that green and purple here are pretty mixed. There doesn't seem to be a separation between these communities in this visualization. And in fact, our statistical analysis was not significant to show a difference between communities on the Keauhou and Kiholo side. So based on this, it would look like there's some connectivity between these two. But I also want to point out the different number of samples in both of these aquifers, right? Quite a few samples in the Keauhou aquifer, and we don't have very many in the Kiholo. So sample size probably has an effect on some of this variability. But just based on what we've seen, the communities in these two aquifers are not different from each other, indicating that there's, across this volcanic rift zone, there may be some water connectivity. The other part of this question was asking about water quality, in particular in the Keauhou aquifer. And just based on some of the maps I showed you earlier, you would hypothesize that there's probably some different inputs of, in this case, nitrogen or other anthropogenic inputs into the Keauhou aquifers. This is just a quick NMDS plot colored by nitrogen, and of the samples that we've collected in both Kiholo and Keauhou. And you can see here that some of the pattern here is separating out between samples that are high in nitrogen versus those that are low. This water quality question is one that we're just starting to get at. I would tell you, this is more of the traditional microbiology. We really haven't even looked in this data as to who is there and what they do. And that really, because we've been focused on trying to answer some of these Ike Wai questions, but that's the next step for us, is to get in and see whether or not we can see differences in microbes, actually who is there in some of these areas. So this is the crest of the data slide that we'll be getting into next, is going to be really great differences between these aquifers. I also wanted to point out, and this is the NMDS plot again from the connectivity questions between Kiholo and the samples from the other aquifers are represented here too. We were using as somewhat of our comparisons, and you can, in our analysis, there are differences in these Waimea and Anai Ho'omalu aquifers as well. They're separating out over here, and so according to our analysis, this connectivity really shows up in this type. When we collected these for comparison sake, but this bottom aquifer also appeared to be very similar to Keauhou, and you can see that's the blue aquifer here, and it's grouping over here with the Kiholo and Keauhou aquifers. So there may be some connectivity in some of these border aquifers, but again, all of these aquifers suffer from really low sample numbers, and so getting at that may be difficult. All right, the last question, the flow paths. Can we differentiate these flow paths distinctly from each other based on microbial community? Here on the flow path identified in the Keauhou aquifer, and so it would be particular blue, green, and those are colored over here in the NMDS plot. Blue is here, green is here, and we then asked whether you can see the blue and the green was statistically different, and the R value supports distinct communities with a little bit of overlap. So blue and green, according to our analysis, are distinctly different flow paths in this aquifer. Blue and red seem to overlap and seem to be very similar. So within the Keauhou aquifer, these two flow paths from source to ocean appear to be different based just on microbial community difference for analysis. Okay, so if we were to look at back at all three of the questions asked, we really could say, based on microbial diversity, we surmised that there was a connection between the high and the low or the basal aquifers based on diversity, but the differences there are being driven by the geochemistry difference between those two aquifers. So that's the high-low divide. The connectivity across the volcanic rift zone between Keauhou and Kiholo, we, based on microbial diversity, saw no difference between those two. So we are proposing that those two aquifers are well connected and that water could flow between the two. And then we also, for the last question, found distinct differences between the blue and the green flow paths in the Keauhou aquifer based on this analysis. So I'll leave you with my microbial mantra. If you just know how to ask them, right? And with that, I'll acknowledge all the different people that have been involved in this work. It's a team effort, right? Everything's a team effort. So Kiana and Kui from our lab, Diamond Brittany, Pam at NELHA, the other Frank Lab members, Sean Cleveland's part of the data group, and he has answered some really dumb questions for me about bioinformatics. So we wouldn't have this data without him. And then just the whole group that is embedded. And with that, I would stop and take questions.
Questions from the audience?
Thank you, Sheree. That was great.
Yeah.
Really cool to hear. So, how much differences in the microbial community does the quality of the water?
Yeah. Yeah, that's a great question. I think we'll be able to do that once we start looking at individuals. Right now we're focused. That's really the next part of. I think we'll hone in on some of those groups that specifically do nutrient cycling, and we'll be able to tell exactly how much is there, how little is there, oxygen levels, all those different kinds of things. I think that's really the big set of next questions that we'll get to.
You had there the
NMDS?
Yes. I'm confused by what the units mean.
Yeah.
But looking at where you get the genetic diversity, it seemed to me not in, you had, and then you was, remind me again.
August and one was November.
Yeah. And the one in August, that's the stars.
The November was stars and the squares were August.
Okay.
Yep.
Can we bring up that, that first plot where you showed us that?
Yeah. First one.
Yeah.
Right? Oops, I think it was right there. Yeah.
It seems like in this one, there are two groups of stars. are consistent all the way through.
Yeah. So
But
you're talking
you didn't
about,
speak about that. What does it mean?
so here or over there? Here?
Here.
This one?
Yeah.
Yeah. No, I agree with you. If you were to just look at this without any other, you would say this is the group here.
Right.
This is the group here, and this might be the third group.
Is when you go to the from the stars to the squares, because those are different seasons. But within the same season, you seem to have two distinct populations.
Yeah. So I would point to you the difference. I think related in this plot, you do see two different populations in the November one. So when you're looking at who's related to who, there's a difference in the November. But once you get to just who's there and not there, you don't see that big of a difference between the. So I would agree with you. There seems to be a separation in these November samples, but it's probably based on phylogenetic related differences, and that could be attributed to you know, the different geochemistry in those wells. And that those other things.
Well, no, and I agree. I think there are differences, but you don't think those differences have any significant meaning at this point?
Well, I wouldn't say that. I would say we're going to pile on top of this. Here, we also have March, and we have, what are the other months we have? And so I don't know that this difference is going to continue to stay that separated.
Well, you could make that statement about all the data you've shown.
Oh, definitely.
Because you've only done a small fraction of what
Yes.
you sampled.
That's exactly right. And so this season out here, I have no idea at this point. All I can say is for the samples that we've collected, so structural patterns in the community structure. Good point. Yeah. And when you look at plots like these, yeah, depending on, you can look at them different ways.
Yeah, so just to add to that, you're asking why are there two different? Basically, we don't know what's driving those two differences, and it's not season, right? So season is
Well, it's because season's not involved in there.
yeah.
Actually, I think these images are a bit obfuscated to the novice viewer like me by merging two seasons together. Because now I'm trying to look just at one season and see if there are patterns, and then just look at the other season and see if there's patterns. And that's a bit difficult for me, who's not used to this kind of data. But whether you know what's driving it or not, it's still it appears to be in existence.
Yeah, it's true. It's there. We don't know what it is yet.
Yeah, and we talked about this because if we continue to see this large difference between seasons, we'll also look just at one collection and look for differences within that compared to looking at all of them together. Yeah.
I'm just an observation. I don't understand it because I don't really try to understand the data. I'm a marine biologist. So.
That's all right. That's a great question. Yeah. Yeah.
When doing the microbial analysis for water quality, are you still going to use these two sets of Bray-Curtis and the Uniprac, or is one going to be more important than the other?
You know, that's a really good question. Right now, we're currently using three. We're using Bray-Curtis, we're using an unweighted Unifrac, and then we're also using a weighted Unifrac, which takes into account abundances as well as phylogenetic relatedness. I think we're probably going to use quite a few of those just to see the differences between and to see what the patterns reveal as far as what we're looking for. I imagine we'll probably use some sequence of all of these as we move forward, especially with, I guess your question was related to nutrients and like who's there. So in conjunction with this, we're going to overlay the concentrations of those nutrients. I think if you remember the slide I showed for the hypothesis where the vectors were overlaid, that's exactly what we're going to try and do. We're going to try and overlay the concentrations of those nutrients within this community structure to see if we see groups of organisms aligned with the geochemistry. So we'll do something similar to this, where we look for communities aligned with the different nutrients that we measure for.
since you have most of the cesspools and wells on the one of the aquifers, what are your thoughts on when you're sampling both of them, especially for the water quality?
Yeah. What are my thoughts about that? The geochemists have quite a bit of geochemistry collected in that aquifer, isotope data as well as source and other things. And I think when we get our microbial data aligned with theirs, I think we're going to learn a lot about how much of that interaction with the groundwater is affecting the water quality of the groundwater, probably. Yeah, I think that's going to be a powerful piece for that aquifer in particular.
Thanks.
Yeah.
Any other questions from the?
Oh, go ahead.
Wow.
I've asked my share.
Because I see the last slide,
The last one?
the summary.
The summary. Okay.
Sorry, I don't know why this is up here. It shouldn't be.
Okay. I lost control, so I'll let you.
No, I'm just trying to get those out of your view. I apologize.
That's all right. I can't forward it anymore.
Oh. Oh, did I do something?
Okay.
Yeah, I took it. I'm
Okay.
sorry.
There we go. Summary slide.
Yeah, I guess, again, questions from expert.
No.
I see a strong connectivity here,
Yeah.
but I see here, things go back. Do we see some kind of contradiction?
Between this one and this one. So this question here, did I put the right slide in there? Oh, you're right. This is the slide that should be in that last one. This is whether or not the green and the blue are distinct from each other. And if you look at, I put the wrong, you pointed that out, I put the same two slides in there. It should be, that last one should be this one, and it's distinction between the blue and the green is what I was trying to show there. Yeah, you're right, I apologize for that.
No, it just shows how this is complicated data. I gotta, oh, I'm sorry.
Thank you so much for your talk. I really enjoyed listening. My question is about how the research results might eventually be applied. Like as we learn more about the flowpaths and connectivity and the microbial communities, how does all this information inform decision making related to like wastewater infrastructure or coral reef management?
Yeah, no, that's a great question. I think the power in this data is really going to be the integration piece with the other pieces, the geophysics and the geochemistry, and does what we see with microbial communities support what Diamond and Henrietta are seeing in the geochemistry about source and discharge out to the ocean. And I would love to be able to have an answer for you at this point, but we're at the point where we now have enough data that we can get together and start talking about it. And I think our goal, from what I understand, is really to drive back to those original questions that came from the community. What does it mean for the aquifer if there's lots of cesspools and nitrogen in the aquifer? And what does that mean for the coral reefs, that are all this water is being discharged to? I'm hoping that I think one of our goals at the end is to come together kind of with this conceptual model of what all this means as far as connectivity, volume, and water quality. I think all of that will come together. I don't know that I have the answer to say about it, but I would say it's coming. Yeah. Yeah.
Sorry. I don't know anything about how groundwater relates. And I'm just wondering, using our flowpaths up here and how related the community is. Do you have a feel for what the rating or perspectives might be and how that might affect them?
Yeah, that's a great question. There's another group. There's a modeling group that is collecting age of water in the same spots that we're doing. So that will get at the residence time. And then the modeling group is also collecting I mean, some of our data hopefully will go into validating those models, but they're collecting, you know, circulation data to try and get at volume. And so that's really a great question, because I think we're trying to get at connectivity, but how that connectivity actually then relates into flow and rates and storage and all of that is really still a very open question, especially for me as a microbiologist. But I think this will go to help kind of validate some of that.
Right, yeah. So I had a similar question to that, basically. Like, so when you say it's connected,
Yeah.
does that mean suppose one little microbe makes it to the other side of the barrier? I don't know how they reproduce, right? But let's say it's two, you know?
Yeah.
And they grow. Is that sufficient to show that connectivity? But does that mean that it was like a little tiny crack in the barrier that just allowed that thing to go? Or, you know, like, if it's connected or not, it's very hard to imagine that the whole, geologically, that whole barrier will not have any crack where some kind of path of water can connect to the other side. What is the volume that is needed
To show connectivity. Yeah.
So let me field this and connect it to Chrissy's question. So our hypothesis is based on the fact that if these units of water are not connected, they're evolutionarily distinct in nature. And so there might be some similarity of communities just to begin with. Each point is representing like between 5,000 and 10,000 different sequences from 2 liters of water, right? So there's a lot of information contained in one of these points. And so our hypothesis is that if they're completely separate, these communities have evolved over time and reproduced, which means like they're gonna reproduce in certain areas. Some species are gonna thrive because those are the geochemical conditions, other species. So from mountain to sea or along the flowpath, the communities are going to change. They're gonna move based on the residence time, based on the geochemistry. But if these are two separate units that have never come into contact with each other, those communities should be evolutionarily distinct, which is why we're using multiple statistical metrics to kind of look at these gradients. So if we're looking at presence absent, like you would think that the best evidence that these communities are not together is if you see different guides, presence absent at different sides. The next layer is if you see different phylogenetically related communities on different sides. But we use the weighted unifrac, which is taking into consideration abundances and relatedness, because that's going to show the gradation through the flow path, right? Because your community is going to change, and your abundances are going to change as well. So we're not saying it's like one static community, but we're trying to make some hypotheses, or trying to make some constraints based on some of these geological hypotheses. And so that doesn't mean that, you know, like there might be a small volume of water circulating that might not actually be meaningful in terms of like water management. We can't answer the volume of water. Like let's say like microbes that are really reproductively, like they reproduce all the time. They're like really good and really successful, productive. Like they're the ones that are going over. I think you're going to see this cross. But the microbial data is really a tool to help us constrain these things, and so we're seeing similar differences, like with all of our different data, it lends to that. But also, like you can have different microbes growing in the same geochemistry. Right? We use the same media to grow up different microbial strains all the time. You can also have similar communities growing on different media. Right? You can grow staph up multiple different ways. So, you know, it's very complicated because biology is very complicated, which is why we apply all these statistics. But hopefully that kind of, yeah.
One or two more.
If the model will be able to identify the amount of water that's close to, would that be helpful in any way?
I mean, we can come up with some new hypotheses and theories. And we have, I mean, we have measurements per volume of water. Like we know how many cells are per volume of water. We can't tell you how fast they reproduce. Because a microbe might reproduce in 20 minutes in its optimal condition, but 48 hours in its non-optimal range. So we don't, we're talking about thousands of different species.
That was my question. Thousands of
Like
species.
tens of thousands, yeah.
So you could have similarities, but degrees of differences that would lead you down the path that they are separate communities. Wow.
Yeah, so many of these dots represent over 20,000 different sequence units. And so that's, to take that amount of data and squish it into a dot that occurs right here, it's overwhelming. When you
Yeah, it is.
Yeah.
It's overwhelming to me, and I haven't even begun to think about doing it.
Yeah.
All right, one more, last one.
The second one, Geophysical data, when you separate out the August and November, like just to me, I know you're gonna have a lot more data layered on top of this, but I didn't realize we were gonna be able to see like maybe August, they're more different from each other than in November.
Yeah, so you're talking about August here, here and here. The August and the November difference just in the Keoho aquifer?
Well, just looking at the Keoho versus Kiholo like similarity,
Yeah.
they're not, or they are similar, but it looks like in August they're less similar than they were in November, or more mixed in
Yeah.
November.
Yeah.
So we're gonna be able to see like that kind of seasonality also.
Yes, yeah, exactly. I mean, what's confounding here too is the number of samples in Kiholo and Keoho. But I think we'll still be able to see, if this seasonality is truly a difference, we should still be able to see that, yeah.
All right, with that, thanks Sheree again. If you haven't done so yet, please sign in the sign-in sheet.
You can probably
Section.
exit it, yeah.
Yeah, I'll take care of it. That's fine.
I just can't even fathom that. I mean, back when I was doing research, I was lucky if I got 20 points.
I know. Yeah, it's overwhelming. Well, I can't process this on my own computer. I have to do it. Yeah.
Take a supercomputer?
Yes,